Hi, my name is Sid Gopalan. I'm a PhD student in Todd Castro's lab, and today I'm going to be talking about an ongoing project that combines aspects of functional and comparative genomics with transposable element biology. So we're interested in understanding how transposable elements, sometimes they're called TEs, how they go from being these typically non-functional silenced elements in our genome to elements that have a very well-defined regulatory role for genes like promoters or enhancers. And we're studying this using the snake venom systems of vipers, which gives us a lot of statistical power to observe genomic events taking place many, many times over because these venom genes are highly duplicated gene arrays in the genomes of snakes. In this system, the snake venom serine protease family in particular, which is abbreviated SVSP, have used transposable element-derived sequences to function as promoters and enhancers. So we were first interested in whether or not SVSP-associated TEs are functionally different from the same TEs that are found elsewhere in the genome. And we find strong evidence that this is the case. We were next interested in understanding if we could replicate these findings in a second species. And so we sequenced and annotated the genome of the Western diamondback battlesnake, or C. atrox, it's a scientific name, and find that the exact same TE family is very prominent in the SVSP cluster again. The next thing we were interested in was to understand the tissue specificity of these functional patterns. And we sequenced chromatin accessibility from skin, skeletal muscle, and pancreas. And we find that this type of functionality is only seen in the venom tissues. And again, this is a pattern that we see replicated in both species. We also generated some single cell multiomic data, which measures gene expression and chromatin accessibility from the same cell, and use that to estimate transcription factors that are most important for SVSP regulation to see if binding differences for these transcription factors seem to be important or not. And lastly, what I'm showing here on the right side are the binding sites for several transcription factors, which show very strong occupancy site bias, which means that they're far more likely to be occupied in one class or versus the other class. And in green, I'm showing the SVSP associated transposable elements, and in red, the non-SVSP associated elements. So again, this is suggesting that these particular binding sites are way more likely to be occupied by transcription factors in the SVSP binding sites. And so this suggests that sequence-based differences are also playing some sort of role in functional divergence. So in conclusion, the regulatory co-option of transposable elements, which was originally described in just a single species, can be observed in a second, more distantly related species. And these patterns of functionality are tissue-specific and likely driven by sequence-based differences. Thank you.